Georg Winter, from CeMM Vienna, speaks about therapeutic innovation through chemical reprogramming of cellular degradation pathways.
So what we're doing in my lab is we're chemical biologists. That means we're trying to navigate that interface between biology and chemistry, which means on the one side we're interested in using small molecules to probe biological systems in order to understand them better. And we like to do that because small molecules have a couple of unique advantages. They have dose proportional effect size, meaning we can dose how much we want to perturb a cell. They're typically acting very fast, so we can measurements very acutely after perturbation, allowing us to approximate causality, also in a cell-based system, which is typically not trivial to do. On the other hand, we're also interested in modulating cellular systems in order to allow us to find new small molecules, new chemistries, and new chemical entities. We do so typically by phenotypic screens, which means we kind of program into a cell what we want the small molecule to do, and then use unbiased large chemical perturbation screens in order to find compounds that do exactly that trick. And so at the heart of what we're doing is really this understand, design, and predict principle. Probably for the most part stick to the understand part today, really because I think this is probably the biggest focus in my lab and really a passion of mine, trying to understand how small molecules interact with native biological systems, aka the cell. And so the particular focus that my research group has at that interface of biology and chemistry is the science of transcription regulation of gene control. We're interested in how cells, particularly tumor cells, turn on and off genes in order to propagate their proliferation, and particularly we would be interested in finding chemical solutions to disrupt these typical gene control programs that go off in cancer cells. And at that interface, we're confronted with a big paradigmatic problem, if you want so, which is the issue of druggability. So some of you might know that there is these proteins that are involved in transcription regulation of gene control, predominantly so-called transcription factors, have for long been considered to be undruggable with small molecules. And I would argue that this sentiment comes for a large part from a very inhibitor-centric mindset that has been prevalent in ligand discovery and drug discovery. What I mean by that is that we look at protein structures, trying to find hydrophobic pockets within a protein that we can where we can fashion small organic molecules that will bind that hydrophobic pocket and block the underlying biochemical activity. The problem is that only around 20% of all proteins in the human proteome would fit that blueprint. And the vast majority of all proteins that are engaged in transcription regulation fall into this other 80% of the human proteome. Meaning, by that inhibitor-centric mindset and inhibitor-centric definition, they would indeed be considered undruggable. So our solution to this problem is simply to change the definition, and we're trying to find small molecules that don't act as antagonists, so they don't block and inhibit, also not agonists, meaning they would bind and activate the target protein, but act as chemical neomorphs. This would describe small molecules that really redirect the function or reprogram the function of a particular protein of interest. And I'll tell you more about that in more practical terms. Still, on a conceptual level, what these chemical neomorphs allow us to do is that they allow us to increase the biological space that we can target with small molecules. In addition to that, they also really allow us to create emergent biology. So think about it almost like synthetic biology, but just using small molecules. We can teach proteins that evolution has not taught them until this point when we confront them with small molecules. So chemical modulation beyond evolutionary constraints. How can we change the function of a protein? We know since now 30 years, this is early work from Crabtree and Schreiber, that when we induce proximity between two proteins, this can be sufficient to prompt a downstream biological consequence. So here, this is a synthetic ligand that is bifunctional, so it has two arms, and with that particular synthetic ligand, they can bind two tagged proteins. So these are two proteins in the cell that are modulated so that they can bind these ligands. And in this particular case, they could show that if we treat cells with this particular ligand. They can induce proximity, and this is sufficient to drive activity of, in this case, a T-cell receptor. So we can do that, and we also have learned through the years that many natural products, so these are small molecules that are generated by nature, act by gluing two proteins together. Think about rapamycin or FK506 that really act as these inducers of dimerization. So the take-home message of that slide is one way and one blueprint to change the function of a protein is to change its interactome, to change the type of proteins that it interacts with in a cellular environment. And conceptually, this is really the blueprint of how we built, how we developed our interest in a new type of small molecule drugs that we call small molecule degraders. I'll tell you more about that, but on the conceptual level, these are small molecules that were initially had, again, two binding elements. One binding element binds to a protein complex or a particular protein called an E3 ubiquitin ligase, and the other part of the small molecule binds to the target protein. This is the target, this is the protein that we want to eliminate chemically. And an E3 ubiquitin ligase is a machinery in a cell. We have around, in the human genome, around 600 of them that it codes for proteins that basically put ubiquitin moieties, these are little post-it marks on target proteins that tells the cellular degradation machinery that is a protein that is misfolded, that is old, that is unusable, and we want to get rid of it. So the idea is here, by using and designing these small molecules, we can redirect the activity of this E3 ligase. We change the interactome of the ligase because we bring into its proximity with a bifunctional molecule a target protein that it typically doesn't recognize, but force the ubiquitination and degradation by machinery that is called the proteasome. Importantly here, we're not talking about tagged proteins. So here we're recognizing endogenous proteins on both ends. So the journey started for me in my postdoc days, where we were building on the heels of this very important discovery from Hiroshi Handa's lab, a Japanese research group, where they identified for the first time the cellular binding partner, the protein binding partner of this very famous molecule that is called thalidomide, that many of you might be aware was introduced in post-war Germany under the brand name Contagan. It was prescribed to pregnant women to prevent morning sickness, and then unfortunately led to the birth of around 10,000 kids with these horrible limb abnormalities. And I think it's one of the most fascinating and darkest chapters in drug discovery. There are many books written on that. I don't have enough detail to go into that, but I would strongly recommend reading them. And I think this was only the starting point for what really then turned out to be a very interesting scientific journey, because these molecules were later on brought back into the clinic because it was realized that that particular molecule, thalidomide, but also more potent analogs thereof, were very effective in killing certain B-cell malignancies, so certain types of blood cancers. At this point, still without our community, neither did they understand how that molecule could cause these limb abnormalities, nor was it understood how derivatives of these molecules could kill B-cell cancers. And so this particular study then provided the first glimpse, because what it revealed is that the cellular binding partner is one of these E3 ubiquitin ligases, a protein called cerebrin. And so what you see here is an experiment that is a so-called chemoproteomics, or a drug affinity chromatography, something that actually I learned while studying with Julia in my PhD. And what that experiment basically entails is that you tether that small molecule via a linker onto beads, and then you pour protein lysates over this bead small molecule matrix and ask the question, what sticks to that particular small molecule? And this is how they identified cerebrin here. And so we then, in my postdoc, surmised, well, if you can use that if you can tether that molecule on beads and use these beads to pull down the protein complex, we might also be able to pull that ligase just to another protein, similarly to have shown it to you on the previous slide. And so we then synthesized this prototypic first heterobifunctional degrader that we called DBET1, and you will recognize that this part of the molecule looks very similar to thalidomide, with a flexible linker here, and here we conjugate it to a small molecule that binds to the bromodomains of a particular type of gene control protein called the BET bromodomain family. So the idea was, with that molecule, we induce the proximity between the E3 ligase, cerebrin, and the target protein B84. And for us, it was really exciting to see that when we now treat leukemia cells, only within 2 hours of treatment with relatively low concentrations of 100 nanomolar, we see near complete degradation of this target protein. And it became even more exciting when we could show that this is not something that only works in tissue culture when we treat isolated cells, but we can also treat animals, so these are tumor-bearing animals, once with 50 milligrams per kilogram of that particular compound DBET1 that was never optimized for in vivo use. Nevertheless, we saw very profound degradation of BAD4. So here you would have to look at the brown dots. This is immunohistochemistry, vehicle-treated tumor treated with the small molecule. And that was also a functional event because MYC, a famous downstream target of BAD4, was then also transcriptionally downregulated. And then there was a lot of excitement generated around this because it's really proof that this is not a chemical biology parlous trick, a Taschenspieler trick, but something that can work in an animal. And if we now jump from 2015 to, I think this is 2023, there's now more than 25 PROTACs in human clinical investigation, most of them for cancer. And I think 24 out of these 25 used the trick that we disclosed in 2015 and co-opt this E3 ligase cereblon by a very similar chemistry. Time will tell how successful this modality will be. I think there are first encouraging signs because 2 PROTACs have received FDA fast track approval targeting a kinase called BTK for refractory leukemias, or targeting estrogen receptor for refractory breast cancers. But if you go back, or if you look at the proteins that you see here, those of you that are trained as biologists will recognize those are all proteins that would in theory be druggable, right? So we do have drugs that target the androgen receptor, we're very good drugs luckily targeting the estrogen receptor, IRAK4 is a kinase, BD4 is a bromodomain. So I think what this modality so far has delivered is another tool to go, a more potent tool to go after well-established drug targets. But this is not what we wanted to do in my lab because there was a second type of small molecule degraders that was increasingly appreciated that are so-called molecular glue degraders. And on a high level, they do exactly the same as PROTACs. They induce the proximity of a target protein, the protein that we want to get rid of, and an E3 ligase. To again cause ubiquitination and degradation by the proteasome. The subtle, though very important difference is that a molecular glue doesn't need to bind to both the target and the ligase, but it typically only binds to the ligase and then it performs what we refer to as chemical surface adaptation. So it changes the surface properties of the ligase, and that is then sufficient to recruit target proteins. And target proteins that have been shown to be degraded or degradable via this molecular glue approach are, for instance, transcription factors, such as IKZF1 and IKZF3. Those are, in fact, those transcription factors that explain why some of these molecular glue degraders, which are these IMiD or thalidomide analogs called lenalidomide and pomalidomide, why they are so efficacious in blood cell malignancies. And I point this out specifically here because these transcription factors are now these truly undruggable targets. So we could not pursue them with a PROTAC, we could not pursue them with monoclonal antibodies because they're intracellular, we could not pursue them with inhibitors because we cannot bind them. They have a spaghetti-like structure and we can't fit a small molecule in there. But we can fit a small molecule into the ligase, change its surface, and then we glue the transcription factor on it. The problem was that these molecular glues up until that point were discovered very serendipitously. So sometimes molecules were studied in depth and then it was realized that we have another molecular glue degrader. And so what my lab did over the last couple of years is really heavily invest in rational approaches to identify such molecular glue degraders at scale. I don't again have enough time to show you all of that, but all of that work is published. And I think there is a couple of challenges that my lab likes to tackle now. One is a more rational design. So we have so far worked on screening approaches that allow us to more rationally identify them, but the next evolution would be also to design them from scratch, and a lot of the beautiful work with making structure predictions and predicting how two proteins could interact if there's complementary surfaces on both sides will enable that work. We're also interested in going beyond reprogramming E3 ligases. So most of the work that my lab has done so far is really focused on trying to find chemical strategies to reprogram the ubiquitin-proteasome, so this particular E3 ligase, with a goal to eliminate disease-causing proteins. But there's many more cellular processes that we can consider for chemical reprogramming, and transcription regulation is one that I'm particularly interested in. What I want to share with you for the remainder of the talk is a story where, and this is now going a little bit more in depth, where we identified actually a new type of modality that is neither a PROTAC or a molecular glue. And it started with, again, one of the serendipitous discoveries where a group realized that this chemical scaffold called an aerosulfonamide binds to an E3 ubiquitin ligase called DK15. And what people then really wanted to do is to say, well, if we have a small molecule that binds to that ligase, can we generate heterobifunctional degraders? A PROTAC that, again, on the one hand binds to the ligase, and on the other hand binds to the gene control protein called BAD4. And we found that molecule actually in the patent literature, so this is sometimes another very idea-inspiring source outside of the scholarly literature. And we tested it in our cells, and it turns out that this is a very, very potent small molecule. So we can induce the degradation, here again, of BAD4 and its related beta family proteins, already in the picomolar range, meaning these are very, very efficacious, very potent degraders. And I think I'm already short in time, so I need to be fast here. But what I'll share with you on a high level over the next couple of slides is that we really went from one surprise to the next in trying to understand that particular PROTAC-like looking molecule. The first surprise was when we now eliminate genetically that ligase that we know that small molecule is binding to, the compound is just as active. So it does not care whether the ligase that it's binding to is there or not. That doesn't make a whole lot of sense for us. I can skip that. And so we then said, well, if it's not this ligase called DK15 that we know the molecule binds, what ligase could it be? We set up a genetic screen to give us the answer, and we arrived at another ligase that is called DK16. So you might now say, where's the big deal? 15 to 16 doesn't sound that different, but in fact, based on structures, we would have there was no chance that we could say, well, if it's not that, it needs to be that one. So this was really a surprise for us. The next surprise came is that when we now did the biochemistry to say, well, for sure the compound is now binding to DK16, the answer was no. So we are binding chemically to the ligase that we don't need, yet we're not binding to the ligase that we do need functionally. This didn't make a lot of sense to us. Here we did a lot of biochemistry to basically show only that there is a pre-existing affinity between the target that we're degrading, BAD4, and these newly identified DK16 ligases. So these two proteins kind of like to hang out with each other already. And this experiment basically just shows that another discovery that we made in characterizing that molecule is we really need two of these bromodomains. So BAD4 has two potential binding sites for that molecule, and for whatever reason we needed both of them to be present, which is a very different observation from all the other heterobifunctional PROTAC molecules that we have designed and described up until this point. And so nothing made sense until we solved together with Lecture Julie's lab in Dundee the cryo-EM structure of this ternary complex assembly. It's the so-called Easter bunny, and once you see it, you cannot unsee it. But what you basically see here is this is the molecule, and rather than binding in cis, as most PROTACs would do, meaning binding to the ligase and the target, our molecule binds, rather than binding in trans, our molecule binds in cis, meaning it binds to both of these bromodomains. It glues these two bromodomains together, and then this nucleates the interaction between BAD4 and this DK16 ligase. This explains why the molecule has no affinity for DK16. It doesn't need to, it just changes the surface of the target protein so that then this target protein has sufficient affinity to bind to the ligase. We could then show that based on this knowledge, we could rationally improve these degraders. We can now make them to up to 6 or 8 picomolar efficiency. And we could also show that by changing the chemical features that are outward facing, we can also hijack another ligase. So this is, again, a ligase that sounds similar, called DKF11, structurally not really that similar. But together with the notion that we can rationally improve these molecules, but also expand them to other ligases, we felt that this is really differentiated new approach towards targeted protein degradation that we call it intramolecular glue degraders. And I think there is excitement because there's, based on the source that you have, there's between 60 or 80% of all human proteins have at least 2 domains, so they would all be within scope of this new modality. We like the fact that we can have avidity, meaning we don't need high potency to both of these domains because we can bind the protein twice. And already told you that we could show with these first examples that we can really drive the activity of these molecules. So the molecule that I've showed with you called IgG3 is, to our knowledge, the most potent degrader that has been published today. And this has been published earlier this year. So I want to thank people in my lab, in particular, Matthias did most of the work that I shared with you over the last couple of minutes, funding sources, collaboration partners, and finally you for listening to me, and I'm happy to take questions.